Welcome to lesson 5.3 Chemical Reactions in the Mouth. Make sure you have your lesson 5 packet ready to go. We're actually going to start with reviewing a little bit of what you've done up to this point. So in 5.1, we looked at a graph of food molecules and noticed that as complex carbohydrates decreased in the mouth, glucose molecules increased. Then in our reading, we learned that spit contains an enzyme called amylase, and we made some predictions about chemical reactions happening in the mouth. So let's just review what we know. So our mouths produce saliva when we eat. Saliva is a mixture of different substances, but 98% of it is water. And as we read in the article, one of the other substances in saliva is amylase. What else we know? Scientists have isolated this substance, amylase, so that it can be used in digestion-related investigations in place of real saliva. We have a small supply of amylase available to plan and conduct our own investigations, so we don't have to use someone's actual spit. So the goal of the investigation is to figure out an answer to the question, do chemical reactions really occur in the mouth? We want to determine whether a substance in saliva called amylase could break down complex carbohydrates down into glucose molecules. In previous investigations, we learned that we can use indicators to test for the presence of complex carbohydrates like starch and the presence of glucose. So, quick refresher, iodine is an indicator for starch, which is a type of complex carbohydrate. If starch is not present, iodine stays brown. If starch is present, iodine turns black or dark purple. Benedict's solution is our indicator for glucose. If glucose is not present, Benedict stays blue. If glucose is present, Benedict's turns orange or dark brown. All right, here's a list of materials that we can use to conduct the investigation. We're going to start with our food sample soaked in water. And we're going to use the same crackers we used in 5.1, and these are complex carbohydrate-based crackers. We're going to use the amylase solution that's representing the chemicals in our saliva. We can use water. We can use Benedict's solution, our first indicator, iodine, another indicator, our 4 microfuge tubes, and pipettes. So, thinking about our experimental design, do chemical reactions really occur in the mouth? Are complex carbohydrates being broken down into glucose in the mouth? What evidence could we collect that would support this happening? So, we should start with a food containing complex carbohydrates, combine the carbohydrate with amylase, that's that chemical found in saliva, and then test for the presence of complex carbohydrates and glucose using indicators. If we start with only complex carbohydrates, we add amylase, and then we test. If I end up with glucose, that tells me my amylase is indeed breaking down my carbohydrates. What else do we need to include? This is an experiment, so we need a control. We want to also combine each food with only water so we can compare that to the amylase tests. All right, on page 6 of your packet, you'll see a chart. This is the top row of the chart. We're going to set up 4 different tubes of materials for our lab. All right, let's start with tube A, and you're going to fill in your pictures as we go. So tube A is going to contain food, water, and iodine. So we're going to add the food to the bottom of the tube, then we're going to add water, and then we're going to add iodine. So think about why we're adding water and what the iodine is going to test for. Tube B, we're going to add the food, then amylase, and then iodine. All right, think about what this condition is testing for. Our next tube will have water, food, water, then Benedict's, food, amylase, and Benedict's. All right, so your tube should all be filled in. Now let's think about our procedure. This is just the order that you're going to put things in the tube. Always add your indicator last. So we're going to add the cracker, then the water, then the iodine, and then we're going to mix the tube. So that's the procedure for tube A. Fill in the appropriate procedure for tube B, C, and D now. All right, now it's time to make some predictions. I have our reminder of what our indicators show. So you're going to tell me what color you think the indicator will be after we mix this tube. So this first tube we added iodine, water, food. What color do you think the iodine will be? Same for tube B, C, and D. Think about, we're starting out with a food that has what? What do we think amylase will do? So take a moment and make your predictions. All right, here are our results. These are the results, okay? So write down the color in your part 5 box, what result color you see. And then in part 6, what does that color mean? What food molecules are present? And I have that little reminder at the top of how to interpret the results. At this point, your entire table 6, all parts should be filled in. If not, pause, go back and make sure you have it all. So we figured out that chemical reactions can happen in the mouth. Complex carbohydrates can be broken down into glucose with the help of a molecule called amylase. So answer the questions on 7 and 8 in your packet. The top of page 7 is a little review of saliva. If you don't remember the answers, flip back to page 3 in your packet and you're reading about saliva. And then you're going to answer the rest of the questions. The first question: do you think that chemical reactions could occur in other places of the digestive system? So we're saying that this chemical reaction is happening in your mouth. Are there chemical reactions happening in other places? Where in the digestive system do you think these chemical reactions could be taking place? Next, what do you predict the old stuff and the new stuff in the chemical reaction would be? So in this lab, the old stuff was complex carbohydrates, the new stuff was glucose. What do you think the old stuff and new stuff would be in other chemical reactions? Hint: look back at your lesson 4 food molecule graphs, or even in this lesson at the graph on page 2. What are some other food molecules that might be involved? And last, this is on page 8, how does what we figured out today help us figure out what might be going on with McKenna? Think about what we've seen as far as food molecules in her digestive system.